The central challenge of computational optical imaging is that we have a multidimensional data space, a data cube, and we'd like to capture it on two-dimensional measurement systems. So we need to think of this measurement system as a coded channel that we encode to get the information we want. There's a great book from last year that was edited by Jinyang Liang on coded optical imaging, how to encode the channel in a variety of different optical systems where we want to capture two-dimensional, three-dimensional images, the light field, temporal images, spectral images, and polarization. And all of those different sensor modalities have their own codes that have been developed for ways to capture the information efficiently. In our last episode, episode 63, we talked about the abstract concept of feature-specific measurement. In the next several episodes, we're going to go through these similar kinds of examples of spectral measurement, temporal measurement, dynamic range, focus, and figure out how to code the channel to capture the data. Our next two episodes will focus in particular on the ideas that are developed by Esteban Vera and his group in the ideas for coded temporal measurement and on coded spectral measurement, the CASI measurements that go back to episode 26. The chapter in Professor Liang's book on CASI was written by Xin Yuan and his group. Here's some figures from their work showing the encoding channel where we try to put each spectral plane on a different code, and then we decode that to recover the spectral image as shown here. And this, again, is images that are drawn from one of Professor Yuan's many publications on this topic, and this one from Liang's book. Now, the balance here is between the physical implementation of sampling and the mathematical sampling structure. As we saw in the last episode, we could develop an optimal mathematical sampling structure, but that may not make sense physically. Here, the CASI system is used because it's a simple way to develop an interleaved spectral code. You could also do that, as shown here, with interleaved thin film filters. The paper on the left is data from this paper from Nature Photonics a couple years ago, showing a Fabry-Perot filters with random spectral sampling, and the paper on the right is from a decade ago or so, where people were looking at different ways to make multispectral filter arrays. These are thinner and more efficient than the coded aperture CASI approach, but much more difficult to build physically. So this is the core challenge, and what we're going to talk about in today's episode is, first of all, what kind of features, what kind of spatial spectral features would be optimal for spectral measurements, and then we'll briefly talk about how we would go about physical design of those kinds of systems. Computational imaging. Episode 64, Spectral Imaging. Here we begin our discussion with a diagram we've seen several times before, thinking about the spatial spectral data cube. So we have a three-dimensional data cube with lambda on this axis and x and y, and these are different ways to sample the data cube. One of the underlying fallacies of this approach is that we probably don't want uniform sampling in the data cube. Why would we sample every voxel necessarily? Is that really the best way to represent and sample the data coming from the optical system? Back in episode 62, we looked at an alternative strategy here with red, green, blue, and clear aperture cameras. This allows us to independently set the focus and exposure of each of the color channels and opens up some freedom in the sampling space. And we saw in that episode that we could take the different images and combine them into an integrated image using a neural estimator. This approach gives us a lot of design freedom and allows us to rethink sampling. In today's episode, we're not going to come up with some definitive best strategy for measuring hyperspectral data cubes, but hopefully it gives you some freedom to think about what might be good strategies and good approaches to evaluating strategies. Ultimately, my personal feeling is that the best approach is something like this image visualized by DALI, where we have a diversity of sensors observing the scene with variable focal lengths, variable spectral response. Many times, for example, you might want kind of a multi-scale sampling where you have broad information over the spectra across broad bandwidth, and then a few very narrow band features sampled to detect some specific analyte or some other feature. But that's all going to be context specific, and the nature of how we're going to sample depends on what we're measuring. The simulations that are presented in today's episode are on the GitHub site under Chapter 10, under the multispectral images, and we look at just a couple of examples of ways to think about sampling based on the principal component analysis strategy that we talked about in episode 63. We begin by looking at this AVERIS data, a very famous data set from Indiana, shot in 1992. If you go to NASA's AVERIS site, you see that the airborne imaging spectrometers have been deployed over the years in many applications by NASA, but for just availability, I guess this Indian Pines data set has been commonly used by a lot of groups just to get a first image of what a spectral image looks like. This AVERIS data from 1992 consisted of 222 bands going from the UV up through the near infrared. So here we see slices of the data cube of a forest in Indiana shot at 500 nanometers, 800, 1100, 1400, 1700, and 2 microns. And you see differences. We're looking at the same data, but for example, here's some feature at 800 nanometers and 1100 nanometers that just looks very different in the blue and in the near infrared. So there's differences in the scene that we can use to analyze, what's forest, what's roadway, what's a planted field, what's an unplanted field, and so on. This is one way to visualize the data, looking at slices in spectra. We can also look at slices in space and spectra. So this is along one of the axes, a slice in space with a slice in spectra. And here you see the spectra look. We have different features, but they're not widely different. They're not like random spectral features, and we see that here when we look at plots just along the spectral axis of randomly drawn points. And you see that first of all, there's transmission windows in the atmosphere in regions where the atmosphere doesn't transmit, so we don't have very much signal where we're in those windows where the atmosphere is not transmitting well. And then within these bands, we see some slight variations. Here, for example, the blue is high here, but still high over there, but low here. The red curve is high here, but low over here. So there are differences in the features that we could use to analyze the underlying structure. As we saw in episode 63, one strategy to look at, to represent the data, we can use many different bases, of course, to represent the data, but a natural basis would be to look at the singular value structure of the underlying data signal and get the principal components. And here's what the principal component spectra looks like when we draw a random selection of pixels to build a matrix of data that we then analyze by singular value decomposition to find the principal components. And what you find is that the vast majority of the variance in the data is explained by just the first few principal components. And so we could represent the data very efficiently by just using those principal components. Of course, there may be some feature that's specifically represented on some higher level principal component, but if what we want to do is represent the data with minimum error, just choosing to sample a few principal components would be optimal. So we can imagine certainly making a spectrometer that just measures principal components. Here's what the principal components look like, and what you see is that the first few loadings, the lowest order principal components, are kind of broad spectral features without much structure. And then as we get into higher order ones, we quickly start to see specific sharp spectral features. And so you could certainly imagine making an efficient camera where, let's say you had a spectral, each pixel is mapped to some of the broad low-level spectral features to give you the general structure of the scene, and then some of the cameras have filters that are matched to these specific sharp spectral features. Again, the underlying challenge is how to design the physical sampling to match what's mathematically attractive sampling, because it actually is possible for us to make a spectrometer with features that are exactly matched to the principal components, let's say on a camera array. But it may be physically easier just to make narrowband filters that would pick up some of these features that we think could combine to approximate the principal components. But certainly the principal component analysis gives us a guiding light for figuring out how to implement optimal sampling. So this is one way to visualize efficient sampling. Let's step back and think about whether this is the only way we could have analyzed sampling on a spectral data cube. And for that, we'll look at a second example here. Srinair's group at Columbia had a study 15, 20 years ago where they looked at spectral sampling with visible images and very helpfully posted multispectral images of these scenes as shown here. This is showing the data in RGB, but it's actually collected over 31 spectral bands across the visible. And we're going to look in particular at this image of beads and analyze the principal components of this scene. Here's an example of some representative spectra just drawn from random points within the beads. Again, when we do principal component analysis, taking as our vectors, sampling vectors consist of individual pixels in their spectra, build up a matrix, and then find the principal components of that matrix, you find that the data is, first of all, well described by just the first few principal components. So instead of making 31 measurements, perhaps we could measure the first 5 or so spectral components and get 99% of the variation of the underlying data. Here's a look at what the first 5 principal components look like from that data. You see, for example, this is something that would sample the blue. This one would sample the green. This principal component samples some blue and some red. The third principal component is mostly red, so we have kind of an RGB filter and then mixtures of our red, green, and blue in the higher order principal components. So we could build an efficient spectral imaging system that had its filters designed to match these principal components, and it would give us the underlying structure of the data. But our assumption that our measurement vector is just consisting of the pixel spectra is an arbitrary assumption, that we could also think of the image itself. The image is consisting of our sample vectors. We could think of, let's take a group of pixels like we did in episode 63. We took a group of 16 by 16 pixels and formed a vector that we used to find principal components. Here we could take a three-dimensional vector, for example, a slice in X, Y, and lambda, think of that as a measurement and get statistics that have spatial spectral features. In the example notebook that's posted on GitHub, this is done by taking one-dimensional slices. So we take a slice along X and lambda and think of that as a vector. And in this case, we have 16 pixels in space and then 31 spectral channels. So we get a 16 by 31 feature vector that we can build up a matrix of measurements from the image data, and then find the principal components of that. And then once again, we find that the principal component spectra is such that, maybe the first 5 or so principal components, or in this case, because spatial spectral sort of patterns, maybe we have to go out to 10 or 15 spectral features out of the 500 or so features in the image vector to describe all the variations. So we could keep, let's say, the first 15 spatial spectral features, and we've compressed the underlying pixel density by a factor of 30. But we have a good description of the variation. But the interesting thing here is to look at, well, what is the structure of the principal components looks like. And you see something kind of interesting here that, again, what you're looking for efficient sampling would not be uniform sampling. So the lowest order principal component is just measuring spectra, basically, no spatial variation at all, just measuring sort of overall spectral features of the scene. And then some of the higher order principal components come in, have very little spectral variation, and just measure the spatial structure of the scene. So a optimal sampling strategy based on the efficiency of looking at the underlying structure of the data, maybe this kind of multi-scale approach where we have coarse spectral resolution and with fine spatial resolution and coarse spatial resolution with fine spectral resolution, and we can combine those kind of features to get optimal sampling. So as we go back and think about what does this mean for measurement design, it means that we have to be imaginative, that certainly it's not going to be efficient or attractive for us just to blindly measure every point in an arbitrarily defined optical data cube. We have to think about how could we sample efficiently in ways that allow us to get the data cube. One way to do this, again, is with array cameras. Here's a paper where we use the uniform sampling of different spectral features on an array camera. And this is interesting. And then, of course, you put this into a neural processor to recover the original underlying spatial spectral image. It would be better not to use uniform focal lengths and uniform camera resolution, or even uniform spectral features. It would be better if the filters had multi-scale structure and the cameras have multi-scale structure. And that's the underlying idea presented in this image that's in the textbook showing the idea that we can have fine spatial sampling and, of course, spatial spectral sampling in a variety of structures. And this may be best implemented in some kind of sensor array architecture. But the point of today's discussion is not to say, here's some definitive best way to go out and measure hyperspectral images. It's just to alert you to the possibility that one can be very imaginative about sampling the optical data cube. We've gone through a lot in these lectures in getting the ability to physically describe the field, how we sample the field, and how we estimate the image. The point of our discussion here in Chapter 10 is just to say, as we design systems for specific imaging applications, we should be imaginative about our sampling structure. The course that the point that we talked about in the introduction with coding for optical sampling mentioned the fact that this is true for spatial spectral sampling, spatial temporal sampling, polarization sampling, and so on. We're going to go through some examples around that. Our next episode will consider video sampling. Here we show again a clip that we showed in episode 26 of the blink of an eye, 133 frames recovered from a single coded frame. So we'll build on the ideas of Jin Liang's book on coded sampling in discussing video capture in our next episode.